Hi, everyone. Thanks for the introduction. And yes, my name is Jordan, and I'm really thrilled to tell you about the work that I did as a Hertz Fellow on how we can create new genomic medicines with man-made evolution. So, as you all heard this morning from Feng, CRISPR-Cas9 is a really powerful method in basic biology and in medicine. And the reason it's so powerful is because you can direct this Cas9 protein in gray to any spot you want in the genome with this RNA molecule in green, and you can generate a double-stranded DNA break wherever you want in the human genome. And this has led to a slew of therapeutics. Most notably, patients have actually been treated for sickle cell disease with Cas9 cutting. So Cas9 has already made a huge impact in medicine. But the problem is that for many other genetic diseases that are not sickle cell disease, Tay-Sachs, cystic fibrosis, a bunch of neurodegenerative diseases, cutting alone is not enough. Instead, what we would like to do is to take a gene that has, a mutation that is leading to a genetic disease, and instead of just cutting it, we would like to precisely replace it with the sequence that most people have, with the healthy sequence. And we can't do that always with cutting alone. So to solve this problem, in 2019, the Lu Lab developed prime editing, and you'll recognize a few of the components of this prime editing system. We're still using Cas9, but it's a little bit different, and we've glued it to this other enzyme, this reverse transcriptase in purple. And we're still using a guide RNA in green, but we've added some extra spinach to the end of it. And when you use a prime editing system, what happens is you still have targeting to a spot you want in the genome, but instead of making a double-stranded break, you can just cut one of the two strands, and then the extra stuff that we've added starts to do its thing. This reverse transcriptase enzyme and the extra RNA we've added engages with that nicked genomic DNA strand, and the reverse transcriptase starts adding new DNA bases directly into the genome, and we can template anything we want with this extra sequence on the guide RNA. And through a series of cellular repair steps, we can actually do what we want to do for most genetic diseases. We can replace sequences of DNA and directly rewrite other sequences that we want in instead. And this technique has been used successfully in all sorts of cell types and living organisms. So what have I been trying to do with it? Well, if you think about it, we've made a very Frankenstein-type protein here, right? We've glued this bacterial Cas9 onto this other enzyme from a mouse virus, and we're asking them to work together while stuck together with a substrate that's, neither one of their native substrates. So this probably could be working better than it currently is right out of the box. And then another thing that was mentioned this morning is that there's precious few ways to deliver these things into the human body to actually use them for therapeutics. And this complex I've described to you is huge. It's way too big to fit into most delivery systems. So ideally what we would want is we want a more active enzyme and we want a smaller enzyme, ideally something that is both active and small. And it's kind of a shame for our purposes that these things evolved to do their native function instead of evolving to do genome editing. It would be great if we could turn back the clock and evolve them for what we want instead. And we kind of can do that. So using a technique developed actually by Hertz Fellow Kevin Esfelt, what I've done is I've modified the system to evolve prime editors for genome editing. So I won't go into the details in the interest of time, but what I've done is I've engineered phage, so viruses that infect bacteria, and I have made these phage only be able to survive and propagate in their bacteria hosts if they can perform prime editing. So if you have a phage encoding a prime editor that works really well, it'll propagate a lot, and if you have one that doesn't do so well, it will eventually get washed out because this whole system is in a continuous flow apparatus. So you have to constantly be prime editing and propagating in order to survive. So it's kind of Darwin in a test tube. And when we do this, we evolve prime editors in the system. We can put in small ones that we want to work better. We get out some things that are pretty interesting. So when we use them in a situation that we're interested in, so maybe doing this AAV packaging of the prime editing system and delivering it to the mouse brain, we can see how these things do. And when we sequence the DNA 3 weeks later from the mouse brain, we can see that before evolution on the left, the prime editor is not doing so well. But if you look at these pace-evolved prime editors, they're installing this LOXP sequence about 50% of the time in cells in the brain, which is higher than most therapeutic applications need. So what do I hope the story gets across to you? I think a really cool thing that's going to come across from the biology talks and the chem bio talks today is that there are starting points from nature that we can use to make new biotech and new medicines. And one of the many ways to do this is with directed evolution, many techniques of which were developed by and are being improved and applied by Hertz Fellows. So with that, I would like to thank the Hertz Foundation for really being extremely supportive of me and of this project, as well as my advisor, David, and then all of my co-authors on this paper, especially Smriti. And with that, I can take questions.
Very cool stuff. It seems like your edit that you put in is a LOXP site, which
Yep.
then you could add yet another protein that is also not meant to do this to then insert more stuff into the genome. Have you
Yes.
tried evolving with that on top of it?
I have not evolved that enzyme. So what he's referring to is that we've inserted this fairly long sequence for prime editors, which is why we needed them to be so active. So this site is a LOXP site, and it has kind of a cognate enzyme that can insert whole genes into that sequence. So in theory, this lays the ground for targeted whole gene insertion in living organisms, but we're not quite there yet. We got to do that second step.
Can you customize the lagoon in any way that would make this potentially more potent or more active within the environment where this should act in the final destination?
That's a great question. So the thing that's been most relevant for this project is we'd love it to be in a mammalian cell, not in a bacteria. And there are other labs that have developed similar techniques that we've tried with varying degrees of success between them. But I think that tech is coming, and I'm really excited for it. Thank you.